Okay, so I think we can get started. Welcome everybody, and thank you for joining us again for another round of the online cell size and growth seminar series. Today we're very lucky to have two speakers who are going to talk from different perspectives about quantitative control over gene expression and protein production, how this relates to different aspects of growth and cellular homeostasis. So without any further ado, we're going to start with Andrew from the Slavov lab at Northeastern. So Andrew, would you like to share your slides and get us started?
Yep. Let's see. All right, how do I look? Okay,
Looks great. Ready when you are.
cool. Great. So yeah, I'm thanks for the introduction, Matt. I'm Andrew Leduc. I'm a postdoc in the Slavov lab at Northeastern University. And today I'm going to be telling you a bit about some of our work on the kind of small vignette of some of my recent work on how cell growth relates to the regulation of protein abundances across tissues and single cells. So when we started on a project recently to look kind of very broadly at how protein abundances are regulated, and here when we're talking about how protein abundances are regulated, we're talking about within a cell type, why does a given protein have a certain number of copies compared to another protein? And at like a pretty high level of abstraction, you can kind of attribute this to a few mechanisms really broadly. One would be kind of transcription. How efficiently is a given mRNA translated? How fast is a protein cleared from the cell? And I guess how also long do those mRNAs persist inside of the cell? And kind of maybe a narrower, slightly more well-defined version of this question are what are the relative contributions of these four processes to shaping the overall proteome of a cell? And so one of the first studies that was pretty influential a while ago that set out to tackle this question was a study from the lab of Matthias Selbach, where they ultimately aimed at kind of answering the question I just laid out. And the way they set out to do this was they simply just measured across the genome rates of mRNA synthesis, mRNA clearance, protein synthesis, protein clearance, and protein abundance. And so the way they were able to make these measurements is due to this metabolic labeling approach, where you can take, if you're doing like mass spectrometry measurements of proteins, or you can do 4AU labeling or something at the transcript level, or essentially you just have a pool of cells. You can do this in vitro, and at a certain time, you can exchange the media with heavy isotopes if you're doing mass spectrometry, and then you can kind of watch the incorporation of these isotopes over time. And these patterns of incorporation allow you to predict, to infer the half-life of the protein, the given protein that you're looking at. And so they set up a relatively simple model, and they kind of just took a kind of straightforward stab at answering this question, where they were measuring the rates of translation and for all sorts of different proteins as well as protein abundance, and they just kind of regressed here. They're regressing kind of the amount of translation against the protein abundance and trying to kind of ascertain some overall dependence. And they can do this for each of these kind of four processes. And the takeaway from the results of this paper, which was again just from 2011, so it's a while ago now, but the ultimate takeaway from this influential paper was that transcription plays a fairly large role, maybe accounting for 40% of the variance, and protein translation plays also a really important role, accounting for almost 50% of the variance. And they had protein degradation, accounting for a quite small amount of the kind of total reason why a given protein might be more or less abundant. But when I kind of came across this result pretty early in my PhD, I always found it quite strange, because if you look at the protein half-lives that they inferred, the distribution is actually pretty broad. I mean, we have almost four orders of dynamic range here, where protein can be, degraded at a half-life as short as 1 hour, and where other proteins can persist for kind of thousands of hours in the cell based on their own measurements. So there always seemed to be something at odds here, because I assumed that this would be a pretty strong force in controlling protein levels. If a protein's stable for thousands of hours, once you produce it, you'd expect it to kind of last and accumulate inside of the cell. And to kind of understand what's going on here, I think it's maybe important to look at the cells that they studied. So they performed these measurements just in kind of cancer cell lines, HeLa cells in a dish that probably are all familiar with. And these cells double about every 17 hours. And so, maybe one reason to explain this result is that actually, when we're talking about how proteins are cleared from the cell, protein degradation and secretion are not kind of the only mechanism here. We also have, as cells grow and proliferate, their volume increases, and the result of this is that proteins are also kind of diluted out due to this rate of cell growth. And here, if we're looking at a kind of average doubling time of 17 hours, this is much shorter than the average protein half-life that even they inferred of about 50 hours. So, it could be the case that in this specific context, maybe we just find an instance where protein dilution is kind of eclipsing this, variation in rates that are specific to individual proteins that come from differences in the rates that they're degraded or secreted. So we want to explore this a little bit more by examining systems that might have a different set of growth rates. And, where can we find growth rates that substantially differ from HeLa cells doubling about every day in a dish? And a really obvious example is kind of the human body or, mammalian tissues where most cell types tend to be, term post-mitotic and terminally differentiated, and we get kind of really varied amounts of cellular turnover. Obviously the brain is a good example where neurons are persisting almost the entire life of an organism, and we have variation down to kind of maybe the bone marrow where cells tend to differentiate and divide pretty fast to provide a new influx of blood cells or immune cells that's kind of needed to keep organisms chugging along. So to get these estimates from tissues, we were able to perform these in vivo metabolic labeling proteomics experiments in mice. And we took a few mice and we pulsed them with these heavy isotopes for either 5 or 10 days. And then we just took a couple of tissues, harvested a couple of tissues from the mice and performed proteomic analysis. And we're able to measure like protein concentrations and degradation rates for almost over 10,000 proteins. And the results of this analysis we found to be pretty interesting and kind of starkly contrasting the results that we were seeing earlier in the paper from Matthias Selbach's lab, where if we're looking at the brain, we actually see that protein clearance rates have a fairly strong influence on the abundance of proteins. So here, we see proteins who are, degrading much faster, having much lower concentrations. And so this kind of strong dependence was surprising to us. And actually, we reasoned that this might make sense in the context of the other tissues that we're measuring. So when we looked at kind of the average population cell cycle time of the tissue and kind of plotted that just against the variance explained by protein degradation, we were kind of excited to see a strong trend here, which was maybe giving some credence to our hypothesis that really the rate of cellular turnover is going to play a strong influence on how much protein degradation rates of the degradation rates of different proteins matter to determining the protein abundance inside the cell. So you might wonder here, how did we really measure these cellular growth rates? And ultimately, we were using just kind of the bulk histone turnover with the assumption that histones are, turning over relatively slowly, and maybe the turnover of the histone can provide some kind of proxy for the overall cellular turnover for these tissues. But I got some good feedback from, I think Mike Lance is maybe on this call, who suggested that maybe this is maybe not the most exact method to measure the turnover rates of these tissues. So, our lab also does a lot of single cell proteomics. So I thought that maybe single cell measurements could give us a better way to kind of more cleanly estimate these growth rates. So we also perform these measurements in single cells here, telling you about an experimental design where we pulsed, again, mice for 5 or 10 days. And so we measured the proteins, and we also took RNA sequencing measurements from kind of paired mice in the same tissue. Here we were just measuring tracheal epithelial cells, but ultimately the data set gave us about a large number of cells at the RNA level and about 4,000 cells at the protein level, with roughly 2,000 shared proteins quantified, genes quantified between these two data sets. And here's just a brief snapshot of what some of the data looks like. You can see that we're capturing a kind of highly overlapping set of cell types, which is as expected, as they're kind of coming from tissues that are shared and dissociated the same way. And I just wanted to include this in here because it might be of some interest to this community. One of the kind of quality control steps we have in our proteomics pipeline is to just compare the total amount of protein measured to the volume of the cell. And these volume measurements come from image estimates of the cells that we make when we're doing the sample preparation. So here you can see there's a fairly strong correspondence between the size of the cell and the total protein that we measure. Similarly, these were kind of from small primary cells. And actually, if you're using kind of cancer cells where the cells are a bit bigger, the measurements on the imaging side for the platform we're using get a bit, the kind of errors get smaller as the cell diameters get bigger. So we're able to kind of measure that relationship quite accurately in many cases. Some folks might say on this call that maybe it's the amount of histone and DNA to the cell volume that's maybe more important, but I just kind of wanted to flash this for people interested. And so back to this kind of growth rate question, how do single cell measurements how might they help us better infer the growth rate of different cell types within a tissue? So here what you're looking at is the kind of heavy to light turnover ratios for basal cells, which is one of the populations that we measured for histone 2b. And what we saw when we looked at these distributions was kind of encouraging, mainly that we get these really kind of discrete populations of cells here. And what's happening here is that you have cells that have not divided yet, where the majority of their histones are light. So they have a kind of this really low ratio of heavy to light histones. And then we have this population of cells kind of around 0 on the log scale or kind of 50-50 light heavy. And these are corresponding to cells which have gone through kind of one incorporation of they've kind of divided and doubled the histones after we did this pulse. And then we have kind of some further distributions for cells that have divided multiple times. And from these distributions, we're actually able to infer the growth rates of all the different cell types that we measured in our experiment. And so we actually have quite a bit of variation here from some immune cells that are doubling on the faster side, all the way down to these kind of mesenchymal chondrocyte cells that are kind of, really very terminally differentiated and not dividing. And so, based on now that we have these kind of growth rates and turnover measurements for all these different cell types, we can look at how the growth rate impacts the relationship between the protein clearance rates for cells that are kind of have a high growth differential. So maybe basal cells, which are dividing faster compared to the chondrocytes. And so you can see when there's a high growth differential, we kind of have a flattening or kind of a reduction in variance of these degradation rates for the cells that are dividing faster, which leads to the shallow slope. And then as we start to compare cell types that have more similar growth rates, we see this kind of slope approaching 1, indicating that the influence of growth is comparable across these two cell types. And another way to frame that is ultimately the dynamic range of these clearance rates is essentially, proportional, inversely proportional to the rate of growth. So essentially for cells that are growing slowly, there's a much larger difference in accumulation of that protein between the most long-lived and the most short-lived. And that's because, again, they don't have that kind of growth, which is applied equally to all proteins that kind of flattens out this distribution. And so if we look at a cell type that, again, has kind of low growth, and we compare how those protein turnover rates or degradation rates correspond to the protein abundances, again, we see this strong inverse trend, which we saw in the brain and we saw in some of these other tissues that we measure in bulk. And we can also look at this across, and again, now we can look at this kind of percent variance explained by cell protein degradation across all the cell types and corresponding to their growth rates. And again, we see this kind of inverse relationship. I think earlier I was plotting against the kind of cellular half-life, so it looked like a positive correlation. It's the same thing here, but I just have growth rate on the x-axis. And so we saw this trend, and this was kind of in line with our other estimates in tissues, where we feel like we had a better handle on measuring these cellular turnover rates within the tissues. So one of the questions we next asked was, is there some mechanism that's kind of compensating and making up for the kind of variability in the proteome when degradation is providing less variation? And so we also looked at the relationship of mRNA abundances to how much variation mRNA abundance explains as a function of growth rate, and we didn't see a strong trend. Interestingly, we saw somewhat of a significant trend for translation, where cells that are kind of growing faster seem to have more control via translational regulation. And this, again, is somewhat in alignment with those. It kind of contextualizes those early results from the Schwanhauser et al. Paper from Matthias Selbach's lab, where they found really strong translational control in cells, in cancer cells that were kind of dividing really fast. So we found this to be kind of interesting and add some perspective for us. Um, And so just to give one last little bit of information about our paper. So what about when, a given cell changes its growth rate? And so, these trends might be interesting, but, they might not give you some specific insight onto how cells kind of regulate their proteomes on kind of shorter timescales or in response to some sort of perturbation or something like this. And so, yeah, these cell types in the experiments I've showed are really quite different. So we wanted to look at something that was potentially slightly more well controlled. And so for this, we kind of looked in the literature and we found some proteomics data sets of some B cells that were first in the resting state, where they are kind of really slowly growing. Maybe they divide once every 6 months. And then in the activated state, where you add a stimulus and all of a sudden they start rapidly proliferating kind of on the rate of once every 24 hours. Um, and so, the difference between these two states is going to be a considerable differential in the growth rate. And so here we just have the clearance rates for the resting state, where you can see that this distribution is pretty broad, right? There's the proteins that degrade the most slowly, which are all the way down here, and then you have other proteins that are degrading much faster. And when you look at this trend, so this curve comes from degradation rates that were measured at the resting state. And if we just kind of tack on this growth and assume what this curve would look like for cells that maybe they're keeping their degradation rate same because it's a similar cell type, but ultimately it's just starting to dilute proteins much faster. Well, what you get is that this kind of curve starts to really flatten. And for proteins that degrade longer, there's kind of this more significant differential in overall clearance between proteins that kind of degrade more slowly versus the resting and activated state when that growth term is kind of added. And so, the result of this could manifest in kind of two scenarios. One, where ultimately the protein levels don't, you do have this kind of reduction in the ability to kind of differentially clear proteins, but maybe the cell translationally compensates. And so one really clear, nice example of this is histone proteins, right? So histones live very long, but they're not kind of totally diluted out when the cell starts growing. Of course, you make histones exactly in accordance with the rate at which these cells are kind of growing. So there's kind of a relationship there between the growth rate and the production of histones. Um, and the other scenario is that there actually is quite a strong trend. So proteins that are really long-lived start to become diluted once the cells start picking up and growing more fast. And so this translational compensation, I mean, this has been argued kind of by a few other groups. Some of the authors from that original paper from the Selbach lab, and other authors who looked at fibroblasts when they went upon growth arrest. And so people have kind of more argued for this translational compensation approach to kind of maintaining the same proteome. Um, and so we wanted to look at this really clearly. So here on the y-axis, we're showing the differential um, in protein synthesis rates on the left-hand side between these. So how do these protein synthesis rates change between the activated and resting B cells? And what we saw was that there's not such a strong relationship to the half-life. So it doesn't seem like they're kind of doing this compensation in such a significant way, where proteins that are longer lived need much more translation to kind of make up for this trend. You can see the histones as kind of clear outliers, right? So the histones have quite a long half-life, but of course they need to be kind of have a large differential in synthesis rates between the kind of resting and activated B cells to maintain that histone level kind of proportional to the amount of DNA. And when we looked at the kind of change in protein clearance, which again is really just coming from the difference in growth from those curves I showed earlier, essentially where you have that broad distribution, then ultimately flattening with a bigger differential for more slowly growing proteins. And we look at the difference in protein concentrations between the resting cell type and the dividing cell type. We see that this kind of increased dilution plays a pretty significant role in regulating these protein levels, more so almost than the kind of specific compensation that's occurring in a half-life dependent manner. And so we found this kind of to be interesting in suggesting that the overall rate of kind of dilution upon cell growth is kind of a strong force in shaping the proteome in a really half-life dependent manner. And so this was really just a small vignette from two papers that we recently have put out kind of from the end of my PhD slash like early in my postdoc from the Slavov lab. And so there's loads more stuff in here. One of these papers is a bit more focused on this growth angle, and the single cell paper is kind of it's a small part of kind of a larger body of work. So if you're interested to learn more about maybe single cell proteomics and how far it's come, and or maybe our ability to make these turnover measurements in single cells and in complex tissues, you can kind of read about it more here. And with that, yeah, I just want to say thank you to Nikolai, who's been my PhD advisor and also been working with over the last year as a postdoc, some of my collaborators who help with the mouse work at Mass General, and thanks to some of the people who are maybe on this call. I had a really fun conversation at a conference with Matthew here, and to maybe where some of these ideas got really hashed out and crystallized, and to Mike for some feedback on the papers that I sequestered. So. Yeah, I think I had about 20 minutes, so hopefully I'm okay on time, and I'll take some questions if people are interested.
That's great. Thanks very much, Andrew. And as ever, please post any questions that you have in the chat, and then I'll call on people one by one to read out their questions. I wanted to start and just ask you. I'm sure you must have thought about this. Maybe you can tell us how you think the approach handles the effects of autophagy or potentially recycling the isotopes in different tissues. I could imagine that varies between cells at different growth rates and may play a role?
Yeah, that's a great question. So this is a bit like technical and maybe slightly less fun, but especially in the in vivo experiments, controlling for the rate of like the incorporation rate essentially is really important. You really can't measure these things accurately without it. There's been a lot of work kind of prior to work that I've done, where people have, figured out reasonable ways to kind of measure this. It essentially relies on looking at proteins that have kind of peptides where you have two lysine, and from the essentially from the, you might have a lysine that has two heavy incorporated, which would naturally happen after you start. But since there's some kind of buffer period, you have a number of peptides that get produced with one heavy and one light combined. And from that mixture, you can kind of do some math and estimate the kind of how that free pool of available lysine changes over the course of the experiment. We looked at this in the single cell populations, and we thought it was really, a cool, interesting finding that within the same tissue, cells that turn over faster were also having kind of almost a faster incorporation of lysine into their proteomes than the cells that were kind of degrading more slower. And we tried to correct for that. I think it's maybe somewhat obvious across tissues that you'd have differences there. The blood kind of flows at different rates, maybe delivers these things differently. But yeah, we thought it was interesting to see kind of within a tissue, there were differences in this incorporation rates for different cell types as well.
Cool, great. Next up, we have a question from Kiki.
Okay.
You can say.
Why do you quantify cell size using histones in particular? I would imagine that the quantity of histones is closely tied to the quantity of DNA. As far as I understand, this depends on the cell cycle stage and may not reflect the overall quantity of biomass of the cell.
Yeah. So let's see. So essentially, with these histones, what I'm referring to, so we're not measuring the cell size via the histones. We're trying to get an estimate of how fast the cells are proliferating for a certain population. So if you have like 100 cells and you've been pulsing for 5 days, and within those 5 days, you can say that, 20 of the 100 cells have divided, then from that, you can kind of back estimate what the original size of the population was. And then from there, you can kind of estimate the growth rate. So we're using the histones to measure really not the size of the cells. We're using them to measure the rate at which the cells are dividing, which we think is the kind of relevant factor that's contributing to the trends that we're seeing here.
If you want to measure division rate, why don't you count the cells at several time points and compare their densities or something like that.
Well, I mean, these cells are right. I mean, these are coming from tissues. So we're making these measurements of from dissociated tissues. Sorry, maybe I have. Yeah, so they're coming from mice, and we were harvesting their tracheal epithelial cells, and then we were, getting a kind of diverse population of cells. So yeah, you definitely can do that if they're in vitro, um, for sure, and that's probably like one of the more accurate ways you can do this. But we also were quite happy that, in cases where, I mean, I'm sure from the literature you could kind of query estimates for the rates of turnover of different cell types, but we were happy to see that we could also estimate these directly from our measurements, and we thought this might be slightly more quantitative than getting kind of ballpark estimate ranges for the different cell types, if that's helpful.
Does this mean that you rely on cells dividing after being taken out of the body and dissociated?
No, so the mice are living, and at day zero, their food sources changed. Maybe I didn't explain this very well. So their food sources changed from the normal food that they eat every day to food that has exclusively heavy lysines, which is an essential amino acid. So they once you start the food being fed, the new proteins that they synthesize are coming from heavy isotopes. And so then we can measure, because we're doing mass spectrometry, as kind of shown in this schematic somewhere here. Yeah. So what you can see is you can measure the incorporation rate. This red would represent the kind of remaining amount of light protein that is slowly leaving the cell, the body of the mice, because you've changed the food source and you're no longer supplying the light. And the heavy is increasing once, and so then we harvest them at 5 days post feeding or 10 days post feeding, and this allows us to, measure the turnover rates that are kind of naturally happening in a minimally perturbative way for the animal. Because, I mean, some people argue that the heavy isotopes maybe slow down some reactions, but as far as we can tell, it's a pretty minimally perturbative way, since it's just an inert, lysine with a couple extra C13s.
Thank you for your patience.
No, no, thanks for helping me clarify that for everyone.
Next up, we have a question from Jan.
Yeah, that was really great, Andrew. Um, I'll look forward to digging through those preprints later.
Thanks.
So one, I guess, question I had was, the changes in protein abundance associated with growth rate that comes from like the kind of just lack of compensation or any kind of feedback control on the protein concentrations.
Right.
Can you rationalize any of that is a way of like maybe supporting terminal differentiation? So
Yep.
like maybe things that support the differentiation of the cell, which is not then now not growing very fast, would be like, I guess you would predict would be very long-lived factors, maybe like the stabilization of a transcription factor or something like that.
Yeah, yeah, totally, totally. I think this is like the exciting angle, which I haven't made like a ton of progress on isolating like specific proteins and saying mechanistically, look, just now as this protein is allowed to accumulate, that is kind of supporting keeping the cell in this terminally differentiated state. I think that's the maybe interesting insight that follows from the initial observation, and I guess obviously ties closely to a lot of the work that your lab has done on finding these kind of relationships. Or
Yeah, we've
uh,
been interested in that, like the size scaling of the
yeah,
concentration and stuff like that.
yeah,
The other,
yeah, totally.
sorry, the other thing I just wanted to mention was I think there's a lot of parallels with your work here and the work from Angelica Ammon on aneuploidy, where they also found like you add, a chromosome to some cell, and then you just generally don't get feedback compensation to maintain protein concentrations. It's just that's relatively rare across the proteome, and mostly you just get proportionally more expression of all of the things on this like extra chromosome that you have.
Right. Yeah, yeah. I mean, that kind of scales with, yeah, that in some ways, the having these kind of complex translational mechanisms that would be in exact accordance to the protein half-life seems a much more kind of convoluted way than the cell potentially evolving proteins that have longer half-lives to be more, useful as the cell is kind of,
slowing
reach
down.
terminally
Yeah.
differentiated state, which I guess would be my vote. But yeah, a lot of the yeah, I'm not sure. I mean, maybe I'm missing some work here, but
Or alternatively, like short-lived proteins would be supporting stemness or something like that. You could look at
yeah,
that as well.
exactly. Exactly. Yeah, that would be yeah, so that would be my interpretation. I've like really wanted to try and make some more concrete statements in this direction, but I haven't quite got there. Um, but yeah,
Cool.
that would definitely be the exciting thing to kind of look at next to find some more mechanistic, we can see that as the cell's growth is arrested, this protein falls, and that's um, yeah,
Yeah. Thank
thanks.
you.
Great. So I think we'll try and do the remaining two questions we have, but we can try and do them relatively quickly. We have a question from David.
Yeah, thanks for the talk. So I wanted to know if you have any thoughts on aging, how aging influences this. You were showing old mice and young mice. We work in yeast, and in the aging yeast, what you see is the yeast is huge. There's a lot of autophagy. The growth rate is very low, like when it ages. So yeah, if you could have any thoughts on that, yeah.
Yeah, so I left. I think that's a remnants of maybe a figure that I haven't edited. The very sad story of the little project is that we had four young mice and four old mice, and the two of our old mice perished at day 9 of their 10-day
Oh,
kind of feeding cycle, which hindered our ability slightly to make comparisons of age. But yeah, we do have a lot to say here. So um, obviously certain cell types might slow their growth rate as they age, which will kind of factor into these trends. Um, but another, I think the kind of more interesting angle that we're have followed up on a little bit is that, you know, protein degradation changing with age is a kind of like pretty canonical, or maybe there's a lot of kind of textbook examples of this. And what we found was that changes in protein degradation with age were impacting proteome significantly, but only in the cell types that were had the lowest growth rate. So, for example, the kind of chondrocytes that we measured that were kind of dividing really slowly. Let's see. Yeah, the chondrocytes that were dividing slowly, they had significant age-related changes in protein expression that were due to changes in protein degradation. Whereas the stem cells, which didn't seem to kind of change their growth rate or too much, where they were kind of still growing fast at old age, they didn't have any kind of protein degradation induced changes in abundance. And so that's the angle that we looked at a bit in the paper. But yeah, I think, as you expand these measurements to other organs and things, you might see that slowdown in cellular turnover will kind of very naturally fit into these trends.
okay. Thank you.
Great. And I think we have one final one from Mike.
Hi, Andrew. My question actually, you overlaps a little bit with what you and Jan were talking about, but I guess the comment that I would have is like if the the idea that the cell would maintain its proteome almost primarily through degrading proteins seems like an extremely wasteful like way of setting your composition. I don't know how you square, like how do you think about that? Is like a half-life, is this like regulated turnover, or is it sort of the useful lifespan of a protein that's determining its half-life? Or yeah,
Yeah, that's a good question. So, I mean, it kind of depends on the way you think of the word regulated. In this case, it would be kind
yeah,
of very soft regulation, right? Where
yeah,
proteins have evolved to have different half-lives for different reasons. And like maybe some are just more energetically stable and others are less stable. And then what we see is that this kind of the stability that has evolved for these different proteins kind of broadcasts itself onto the abundance, which is less of kind of a hard regulation and more of a softer kind of regulation that is just, okay, well, this protein's less stable, therefore there's less of it if the cell is long-lived, kind of relative to the other proteins that are there.
yeah. I mean,
Um,
the correlations are awesome. I'm just saying, it's like I'm trying to think why a cell would want to set its composition by such a wasteful, because it takes a lot of energy to make proteins and degrade
right.
proteins. Yeah.
So if you're
Anyway,
you're saying kind of if a cell is yeah, I mean I think the cells have to kind of do some natural level of protein turnover, even if a cell is terminally differentiated, just to kind of, I guess, make sure that
yeah.
aberrant proteins don't build. And just maybe it's more of a something where yeah, they're not necessarily choosing to do it that way, but it kind of came out from
It's necessity.
the fact that, yeah,
Yeah.
or proteins have different levels of stability. Therefore,
Yeah.
as cells grow more slowly, you kind of, yeah, that would be kind of, again, it's maybe less of a choice. Like, we want less of this protein. Let's figure out how to destabilize it in some kind of clever way, or adding ubiquitin more frequently or something like that. But yeah, I do think there's interesting differences in, like, cell type specific mechanisms of degradation but to me this seems almost like a larger effect in some ways. But we have a lot on differences in protein degradation across tissues that are more slow in both ways. So that yeah, for sure what you said, there's kind of still some regulation more active on the active kind of side where a given protein change, there's a lot going on as well.
All right, fantastic. Well, thank you again, Andrew, for a great talk. I'll ask you to unshare your slides.